The nervous system is the most intricately organized data processing system on Earth. Your brain contains an estimated 100 billion neurons which are specialized for carrying signals from one location to another. The nervous system have two main anatomical divisions, the central nervous system and the peripheral nervous system. A neuron consists of a cell body containing the nucleus and cell organelles, and long thin extension called neuron fibers that convey signals. Most of a neuron's organelles, including its nucleus, are located in the cell body. Arising from the cell body are two types of extensions, numerous dendrites and a single axon. Dendrites are highly branched extensions that receive signals from other neurons and convey this information towards the cell body. Neurons require supporting cells called glia. It intermingles with the essential elements of nervous tissue, especially in the brain, spinal cords, and ganglia. Axons that convey signals are enclosed along most of their length by a thick insulating material called the myelin sheath that resembles a chain of oblong beads. Each bead is actually a Schwann cell, and the myelin sheath is essentially a chain of Schwann cells, each wrapped many times around the axon. The spaces between the Schwann cells are called nodes of Ranvier, and they are the only points along the axon that are leaky, requiring nerve signals to be regenerated. Everywhere else, the myelin sheath insulates the axon, preserving the signal. Thus, when a nerve signal travels along the myelated axon, it appears to jump from point to point as it is rejuvenated at each node. The resulting signal is much faster than one that needs to be regenerated constantly along the length of the axon.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membrane. This potential, called resting potential, is always negative inside the cell. It ranges from negative 40 to negative 90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on its stimulus. If potential is made sufficiently less negative, it reaches a level called threshold and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasms with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasms consist mainly of positively charged potassium ions and large negatively charged organic molecules such as protein. Outside the cell, the extracellular fluids consist mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up the cell membranes, they must pass through channel protein that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane traveling to specific proteins called potassium channels. Although sodium channels are also present in stimulated neurons, they remain closed. Since only potassium ions can cross the membrane and potassium ions are more concentrated inside the cell, they are fused out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration difference is balanced by the electrical attraction tending to pull them back inside. This is the point in which neurons reach resting potential. Reaching resting potential on this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside the neuron must leave to create a resting potential of negative 16 millivolts, but it is action potentials, not resting potentials, that carry information through the nervous system.
A stimulus is any factor that causes a nerve signal to be generated.
Examples of stimuli include light, sound, a tap on the knee, or a chemical signal from another neuron. A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated channels to open and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell had diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time, we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium channels rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
In humans, as in a lot of vertebrates, the central nervous system develops the neural tube. By about 25 days after conception, 3 bilaterally symmetric bulges emerge from the head that appear at the end of the neural tube. This eventually developed into the 3 major parts of the brain: the forebrain, midbrain, and hindbrain. At about 40 days, the tube starts to bend to enable growth continue within the cranial cavity. These bends are found in 2 very specific places. By 50 days, the cerebral hemispheres are developing. By 100 days, they are very obvious. In the following months, the cerebral hemispheres expand enormously and cover most of the brain. The forecomplement of neuron is present shortly after birth. The glial cells continue to increase in number until adolescence, at which time the brain becomes fully mature. The structures of the forebrain, midbrain, and hindbrain can be revealed by taking a surgical section through the brain.